Well, hello again, and now we start looking at the synaptic transmission. So again, still in chapter 10 of the nervous system. So I went back, and this is one of the first slides, but I didn't cover it earlier because we're going to talk about synapses more in depth. So I'm bringing this one down. But all a synapse is where neurons communicate with each other. So it's wherever, so this is the axon right here, right? Wherever the axon is and it's leaving, it's going to come down to an area where you've got a synaptic bulb, or what they'll call terminal sometimes, and that's where, again, that's where the neurotransmitter is released. Wherever this bulb is and the cell that it's going to communicate with, this is an area called the synapse. That's what it's showing. Whoop, that was an interesting little part there. That's what this over here is showing. This is the synapse. I've got one cell, one neuron sending a signal on this axon. It gets to where it's going to drop off the message. Now I've got another cell body or whatever, a cell that's going to be receiving it. And that area is called a synapse. In this little picture, this is one neuron talking to another neuron. So wherever the cell body that is sending the signal is called the presynaptic neuron, because it's before the synapse. And the one that's receiving the signal is the postsynaptic neuron, because it is receiving the synapse. So that's the way this works. Now, what we're going to also learn is, in general, especially in the peripheral nervous system, wherever this synapse is, there's going to be kind of a swelling. There will be a lot of different neurons coming in and synapsing. And so in the next chapter, we're going to talk about preganglionic neurons and postganglionic neurons, and they are exactly the same. The synapse, the ganglion, kind of represents where the synapse is. And so you can refer to it either different way. Now, synaptic transmission, again, this is early in some of the slides, but I just wanted to show you this. Synaptic transmission is a one-way transfer of information. So I've got a preganglionic neuron coming down here. This is the synaptic bulb here, and it's going to release the neurotransmitters. And this here is representing the cell body of a postganglionic neuron. That's what it says there. I don't know if you can read it. So again, as it says, the impulse travels down the axon of a preganglionic neuron to the axon terminal. And then when the impulse reaches the synaptic knob or the synaptic terminal, it's going to be calcium ions that come in. All the way down, it is sodium ions that come in. You know, hopefully you've watched the video about how an action potential works. It's sodium that goes into the cell, and it is potassium that leaves the cell. But once it gets here to this area called the synaptic knob, it is then calcium. Boy, that's again, bad little drawing there. It is calcium that's going to enter, and calcium is what's going to cause a release of these neurotransmitters. By exocytosis, and hopefully you know that and don't have to refresh yourself. Exo, outside, cytosis, cell. It is taking things that are inside the cell and dumping them on the outside. So the neurotransmitter will exert either an excitatory or inhibitory effect on this postsynaptic neuron. And what it's saying is, remember, we had that negative 55, which was our threshold. And we had negative 70, which was our normal or resting membrane potential. So we have to get from negative 70 to negative 55. So some of these neurotransmitters, as we say, are excitatory, which means it pushes us closer to negative 55, while the ones that are inhibitory are the ones that are going to be pushing us farther from negative 55. So when we look at it, the synaptic transmission, now we're way down to, I think it's slide 41, the transmission of a nerve impulse from one neuron to another. The released neurotransmitters cross the synaptic cleft and react with specific receptors on the membrane of the postsynaptic neuron. Sorry, still having issues with the whole tooth thing, but I'm trying. So the effects of the neurotransmitter vary, just like we said. Some will open ion channels and others will close ion channels. The ones that open ion channels make it easier, and that's what we called excitatory. The ones that close it make it harder, and that refers to as inhibitory. The chemically gated ion channels respond to neurotransmitters. That's what is going on. The neurotransmitters are chemical. So these local potentials, which we talked about earlier, these are the ones happening on the dendrites and the cell body, are results in changes in chemically gated ion channels that are called synaptic potentials. We have excitatory neurotransmitters and we have inhibitory neurotransmitters. The excitatory neurotransmitters increase the permeability of sodium ions, which as it says, brings the membrane closer to the threshold, makes it easier to generate an impulse. Inhibitory, they are going to move the membrane farther from threshold and makes it more unlikely to create a potential. Hopefully that makes sense. Excitatory makes it easier, inhibitory makes it harder. So we group these in, and again, scientists are terrible about making sure everything's labeled and in groups and sorted and over again. And so hopefully you'll understand this pretty well. I don't want to make fun of it, but they kind of group them in two categories basically as potentials: the excitatory postsynaptic potential and the inhibitory postsynaptic potential. The excitatory is going to make the membranes change in which the neurotransmitter opens sodium channels, which means that sodium comes in, causes depolarization, right? Depolarizes the membrane of the postsynaptic neuron, makes it more likely that I'll have an action potential. Inhibitory is going to be where the membrane changes the neurotransmitter and it opens the potassium channels. And as potassium starts to kind of leak out, what that does, since I'm losing some of the positive that's the little positive that's in the membrane, that means that I'm going to be hyperpolarized, which makes it harder to send a signal. As it says here, hyperpolarizes the membrane, and it makes it less likely that I'm going to have an action potential. This is a picture showing how some of these neurons are. These neurons, again, if we took a typical multipolar neuron that is in your brain, there is over 10,000 in general, 10,000 different neurons communicating with it in different areas. So I've got all of these things represent neurons coming in and dropping off their signals onto this one neuron. So I've got all of these axons coming in from different structures. And then not only that, I'm going to have them coming in on dendrites, and then the dendrites themselves are going to have little receptors that are coming in. So in this one alone, I probably have a dozen or so that I can see on this picture that are communicating with that. Now, some of them are going to be excitatory and some of them are going to be inhibitory. And so as all of this information, again, gathers up around this area called the axonal hillock, we will add them all together, and they have to equal up to a positive 15 millivolt stimulus. Again, I get that number because that's the number that's between negative 55 and negative 70. Right? So we start at negative 70. We have to get to negative 55 to create an impulse. And so if we have all of these, but the overall number value of the inhibitory outweighs the number of the excitatory, nothing will happen. Now, this process of it all coming together is called summation. That is where everything kind of sums together here and hopefully hits that positive 15 to send a signal. So again, as it says, net excitatory effect leads to greater probability of an action potential. Net inhibitory effect does not generate action potentials. So summation of all of the inputs will usually occur at the axonal hillock, and on this it kind of calls it the trigger zone. So hopefully that makes sense in how this is working, right? So wherever an axon goes and releases the neurotransmitter, that is called a synapse. Usually the neurons are talking to each other, and if it is, there's going to be multiple synapses on the cell body of a typical neuron. And that neuron is going to summate all of the information that's coming in. And if I have a net excitatory, which means I'm going to get closer to this positive 15 value to get over the hump, I'm going to have a greater potential to send this action potential. If it is inhibitory is the net, then it's going to be farther away. So again, hopefully that makes sense.